Hello, good evening. It's another biology session. So what are we going to be covering today? Today in the live stream from Taylor Tutors, we're going to be going from A-level biology stuff. Let me just mute myself on YouTube. Okay, so I can see all your comments coming in over here. If you see me just like looking over here, it's not that I'm ignoring camera. I'm just actually paying attention to you lot. I'm also going to be handling, I'll go over some of the comments that our Ultimate Guide subscribers have left on the course. So hi to those of you who are jumping on and saying hello. What I was going to do first is to say, any of you who have used the Taylor Tutors resources, you've checked out the videos, you've done some of the worksheets and template notes and stuff like that. Next week, we're doing a bit of a promo, and there's not going to be any sort of new on offer. You can still use the coupon Try It Free to get 1 month totally for free as long as you start within the month of September. But new to the course, trying it out. And so those people who have been using it, whether that's just been this month, or it may have been that you've been using it, all of the last year, we want some students to volunteer to come and join us in a live. You can either have your video on, or you can have your video off and just join us by audio, but you can join me in the room. Like Ronnie and Rich and I are usually here sort of pinging back and forth. Just for a few minutes, we're going to ask you some questions, what you like about it, what you don't like about it, like anything you can think to improve it, how you use it, just a few basic questions. Nothing's going to like really stress you or tackle you. We're not going to be asking you biology, chemistry, or maths questions. It's only going to be questions about how you use it and what you like about it and things you think we can improve, just to show other students. It's just more genuine when it comes from you lot. Like I know that it's awesome, but people aren't going to believe me necessarily because I wrote my course. I'm also always going to think it's awesome, but you guys have been using it. Hey Kane, hey Amina. You made it for a biology stream. Yes, you did. So obviously, if you're watching me live right now, think about any biology questions. Maybe you've got some little concepts in there that aren't quite clear in your mind. And with biology, the concepts are not too tricky on the whole, generally speaking. Some of them are a little bit tricky, but most of them are quite easy to understand. However, they are, if you don't to actually like remember them. Understanding is so key to the comprehension. Hey Ryan, hey Bumblebee. The mustache is a bit all over the shop today, actually. It's all straggly, it's ridiculously long. It needs crazy mustache wax, and yeah, look at it, it's just out of control. I can basically poke myself in the eye with it. Like, it's, yeah. Hey, Sane. Okay, so well, I'm going to start off with, whilst you guys are thinking of some questions, if you wouldn't mind including your exam board when you submit the question, because obviously the way I'm going to answer it may change. They're very, very similar, and I can give a broad answer that's going to be the same. I've got a whiteboard if I need to use it just over here. But what I wanted to do, first of all, was go to the course, and I'll just give you some of the questions. So Saba asked me, with monohybrid and dihybrid inheritance, are the phenotypic ratios always in monohybrid 3:1 or in dihybrid? This is probably me way easier to draw on a blackboard, but the answer is, if there's nothing weird going on, then yes, that's what's happening. But that's making various assumptions. It's assuming that there's been no linkage, sexual linkage or somal linkage, which will disrupt those regular ratios of phenotypes, which will mean that it's often a way that you're going to have to use in the question. It's like the ratio is not 93 to 3:1, suggest the reason why. And then you'll have to decide, is it actually sex linkage or somal linkage? And by the time you've studied that, it should be a little bit obvious as to which one it is. To draw out the basic 93 to 3:1, the dihybrid cross. I'll do the 93 to 1 to start off with, so I'm not scaring anyone too much. Year 2 photosynthesis is complicated. It certainly is. Oh, I'm going to have issues. I've got 2 monitors today, which is going to help me with the comments because Ronnie and Rich are abandoning me, leaving me all alone to fend for myself. But if I can't, means I can't do my laptop. I might see. Yeah, I'll save up the questions I need to on the whiteboard. I'll do it at the end because I have to disconnect all my cables and whatnot. Oh, Ronnie is there. I'm bad mouthing you, Ronnie. Ronnie, I know you've got ridiculous amounts of maths doing to be here if you don't want to be. But she's such a glutton for punishment, she probably will not go away. Thanks for helping you get the A star and allowing me to get into. So Bumblebee, which university you're at, I want to know. And Bumblebee, do you want to join us next week on Tuesday, Wednesday, or Thursday? You can take your pick. It'll be the same time, 8 o'clock in the evening, 8 till 9, but you'll only be needed for 10, 15 minutes. If you want to come and tell your story, I'd love to hear from you. It'll only take 5 to Rimmer's was year 2 photosynthesis. Okay, so the photosynthesis and the respiration, the beginning of year 2 stuff, is itty bitty, tiny, interlocking details. This combined with it, it's like very detail heavy. The good news is when you can get to the point where you can like just sketch it out and you understand what gets converted to what and why and where it happens, the questions, you've gone into all the detail. There's not much application that they ask too much there. So the good news is that once you've got the photosynthesis and the respiration biochemistry in your head, the questions are really simple. Whereas the other, very easy to get your head around. However, it doesn't necessarily lend itself to good marks on the exam because they're a little bit wishy-washy, lots of suggest, and how would this interaction affect that interaction? And so often you find this sort of double-edged sword. Either the content is tricky and the questions are more straightforward once you get it, or flip reverse so that it's easier to understand and the questions become a little bit more tricky to understand what they mean. How does IAA control tropisms in plants? So IAA, this question from, I don't even know what your name is there. Suga, if you want to give me actually address you by name. So IAA, indoleacetic acid, is an auxin. Auxin is a name for a family of plant hormones, basically growth factors. Different people call them different things. They affect plant growth. Basically, in plants, they can't get up and walk away. So they have to respond to their environment, and that's called a tropism. You have taxes and kineses, which are responses by organisms that can move. So the thing that sets tropisms apart is a tropism is a response by a plant that can't move. So it can't move its physical location, but it can move its stems or its branches or its orientation. IAA is an auxin type of plant hormone. It does different things in shoots and in roots. Let's talk about shoots first. It's nice and easy to get our head around. Let's say we've got our shoot growing parallel to the ground. It's horizontal. And let's pretend the light is coming from straight overhead. Obviously, these circumstances are a little bit models, but you've got the light coming from here. Now, this basically means the IAA is kind of like afraid of the light. It's going to go to the shaded side. The IAA is going to hang out on the underside, in the shaded side. And that's the same for roots and shoots, but it has different effects. In the shoot, cell division here, it's not causing the cells to divide more. What it actually does is weaken the cell wall. It allows it to stretch, basically. And so the cells on the bottom, where the IAA is, it can grow faster, but not divide. It's just cell elongating. So it's going to basically, if the outside wheel of the car turns fast, it's going to start turning towards the light. So IAA is an auxin type of auxin, a specific type that causes cell elongation in shoots. It goes to the shaded side, so the shaded side grows faster and grows towards the light. Now, in roots, does almost, there's some of it's the same, some of it's opposite. In roots, so let's say it's a root growing horizontal to the ground, right? The IA goes to the shaded side. That's exactly the same up until this point. But the impact, the effect of IAA in the roots, it inhibits cell growth or cell division, which means that the cells grow faster on the top side, which means they start growing away from the light and obviously then into the soil. So the effect of auxins or IAA on plant growth is called a tropism because it will respond to the stimulus. In shoots, it will go to the shaded side, cause cell elongation, grow towards the light. In roots, it will go to the shaded side, cause cell will grow faster, and it will grow away from the light. Okay, Ronnie's already talking about maths. It's a Tuesday, Ronnie, not a Thursday. Okay, so you're going to Cambridge. Bing, high five, Bumblebee, for getting into Cambridge. What are you studying? I'd like to know. I mean, if you want to come and join us, then I'd love, it would be great. I love catching up with old students. I had a good chat with an old student yesterday. So yeah, if you want to come and join us, it would be great. If not, then just check in with me. If you'd rather not do it on YouTube Live, I'd love to catch up with you and just see what's going on. You'll be starting term pretty soon. It's all going to be pretty exciting. Let me know if you're a full next week. My first ever lecture was, oh God, actually, I say that with confidence, 4th of October, actually. It was 4th of October, Monday, 4th of October, 2004 was my Professor Coakley, and I sat there and I didn't understand a word of it. Never had any trouble understanding anything in my A-levels, basically, and then get to university. The first lesson was like, oh my God, if it's going to be like this, I'm not going to complete 3 years. Then it turned out he was just a bit of an old, he still wrote on the blackboard. I mean, we had whiteboards everywhere. He was so old school that he was writing on the blackboard, and it was everybody didn't understand, because I think he just wasn't very good at explaining things. Sugar. I don't understand. Ronnie, you said sugar, and I don't understand what it means. Oh, your name, sugar. Gotcha. Can I talk about lactose intolerance and the things surrounding it, like someone how gets it, but also the solutions if someone wants to drink milk but is lactose intolerant? Okay, so all mammals, we are mammals. We have mammary glands, we have nipples, even the boys, which means we feed our young on milk. So every young mammal has the enzyme lactase, because milk contains lactose, the sugar. We need to break it down. Lactose joined by a condensation bond. The reaction to break them apart is a hydrolysis reaction using a specific enzyme, lactase. So that enzyme is present in all young mammals. Over evolutionary time, typically it turns off. We didn't really continue to drink milk into our old ages. Humans, around about 29,000 years ago, this is all outside the spec stuff, you don't need to know, about 29,000 years ago, retained the expression. So we had the gene already, and the gene just stayed switched on for longer, which meant we could drink milk and metabolize it and get energy from it. Tolerance. Some humans, I think that happened, actually, I don't know the first instances, but certainly in, I think, Levant, which is basically sort of the Middle East through to Western Europe, the Cro-Magnon in southern France. I think that's where the lactose enzyme stays switched on the longest, or the earliest occurrences of that. So in East Asia, in China, and lots of Southeast Asian countries, the lactase enzyme, that was never, they had the enzyme, everybody does, but it didn't stay switched on. So it's rarer to have the lactose enzyme alert and in, I actually don't know about South America, to be honest. I've traveled there a lot. I think they do drink milk, so I assume that they have the enzyme switched on as well. So why does it cause problems, first of all? Well, normally, if you can digest lactose, what happens is the enzyme hydrolyzes the glucose and the bond, the glycosidic bond, breaks them apart, and you have two monosaccharides, and then they can be absorbed and you can use them for respiration. If you don't have the enzyme, the molecule stays as a disaccharide. They both stay joined up. This thing goes into your gut, which is then digested by microorganisms in your gut that are normally there completely healthily, doing you no damage. But this extra food that they don't normally have, and when they respire it and you're not respiring it anymore, it's being respired in your gut by the bacteria, usually bacteria in your gut, then that's what causes the diarrhea, is the byproducts of that, which are going to cause all those problems. So solutions. You can buy milk that has been treated with the immobilized enzyme lactase. So they just basically pour milk through a set of beads that have the immobilized enzyme in there. This breaks down the lactose into glucose and galactose. So you basically, it does the sort of the bit that you can't digest, it does for you. I have no idea what it tastes like. Probably tastes a bit sweeter because you've got instead of having one lactose molecule, you've now got two monosaccharide sugars in there. So I actually don't know how it tastes. Lactose treated. I'll be able to tell you, obviously there's lots of milk alternatives. Wheat milk, is there wheat milk? Well, certainly oat and coconut and all sorts of soy, all sorts of types of milk alternatives. what are the other solutions? I don't know. I know with peanut allergies that you can tiny dose yourself and effectively sort of retrain yourself. But I mean, don't do this without medical advice, because if you have a peanut allergy, seek a doctor's advice. I wouldn't want to be giving anyone the idea that they could just start eating peanuts if they have peanut allergy. But I know with some allergies, you can effectively build up a tolerance. I don't know whether that works with the retention of the expression of the lactase enzyme. In the future, I can easily see situations arising whereby you can have a little bit of gene therapy. You have the enzyme in your DNA already. The problem is it's switched off. As an adult, it switches off. So you could just find something, a little transcription factor, which would turn on the gene naturally because you have it, and then you would be able to eat products containing lactose. So I don't know if there's any amazing quick fix for that. Maybe in the future gene therapy, or you're going to have to look at, there's lots and lots of products out there now for people who are lactose intolerant, because it's very common. If you don't have issues with lactose, then there's basically nothing wrong with eating dairy products. But if you are lactose intolerant, it can cause, upset. Ryan's asking Ronnie maths questions because she likes hijacking my sessions. Okay, how I was whittering on for ages. Photosynthesis, FADH, etc. I'm really confused. Okay, this is going to be way easier and probably more effective with a whiteboard. So I think I'm just going to cave in. Ronnie, are you set to be here? Because if I start writing on the whiteboard, I'm basically going to have. Let me see if I can arrange my screens in such a way I can still see the comments and write on the whiteboard at the same time. So let's try something like this. Okay, that's a better arrangement. I can probably do that. Smaller, so if I disconnect this, hopefully it won't take me offline. Wonderful. Okay, so I might need to go and get a cloth. Okay, so this whole setup needs tinkering with. How hot is this light bulb going to be? It's a low energy bulb. Let's try taking that out and flicking this light on instead. Oh, it's still super dark. Probably better than crazy reflection. I think we'll do. It's my next job to get all my lighting sorted out. Okay, NAD and FAD and FADs and whatnot. These are all called coenzymes. So I'm just going to put on here, how high can I go? Somewhere like here. And what do? They are molecules that they are in your system already. You don't really make them very often. They are sort of just recycled, and they all have two different forms. They can either grab hydrogen or they can let go of hydrogen. So they can move hydrogen because they can either have it or they can let go of it quite happily. So we've got NAD, NADP. You only come across NADP in photosynthesis, so you can think of the P as sort of tagging you to photosynthesis. You also have coenzyme A. I'm not going to write about that because you don't really, it's pretty straightforward where that comes from. Okay, these are all converted, or can, if they want to, they can accept a hydrogen atom. So when NAD grabs a hydrogen atom, it becomes NADH, and this is the reduced form over here. And the opposite of reduction is oxidation. This is the oxidized form over here. So we're just going to add an H, literally add an H to these. technically you can write NADH2 and stuff like that, carries more than one, but the mark schemes always just refer to them as these. I always use the terminology that's in the mark schemes. Okay, so this is a reduction. Well, these are reduced NADH, reduced NADPH, and reduced FADH. And we can also go in the opposite direction. Right. So basically what happens at various stages of respiration, photosynthesis, and different ones work in different reactions, but you're going to have a third reaction going on over here. Let's go. What did you ask me about? You asked me about photosynthesis, or did you ask me about respiration? I'll use the context of the one you gave me. Too many comments. I can't find it. Photosynthesis. Okay. So in photosynthesis, oh, it's less obvious in photosynthesis. Let's draw it. Okay, so this is going to be like light-dependent reaction 101 in like no seconds flat. You have light energy. In fact, let's draw red. Red's kind of a bit more lighty. Strikes the photosystem. This is called photosystem 2, rather confusingly. Too many pens. Drawn with Roman numerals, not the number, and this is photosystem 1. They discovered this one first. It's not chronologically first, but they discovered it first, which is why it's called photosystem 1 and then photosystem 2. This basically excites an electron from the chlorophyll. So the energy, the photon strikes an electron, the electron goes, woo, I've got loads of energy. Jumps up, gets excited, it literally gets buzzing with energy. It then flows through a membrane back into the chlorophyll in photosystem 1. It grabs more light energy, which shoots the electron out of the photosystem entirely. The energy that's lost here is used to pump protons. So there's loads of protons here, and they're pumped into here. So this is a hydrogen ion. It's just a proton. A hydrogen ion and a proton, 2 words for the same thing. These are going to diffuse over here, so we have loads of protons. We build up protons here. It's called a proton gradient. Lots of protons here, not very many protons here. So you have a concentration gradient. This allows the protons to diffuse through. So now we've got a proton and an electron. Well, this is going to form, what have we got? Basically a hydrogen atom. So a hydrogen atom, 1 proton, 1 electron. That's what a hydrogen atom is. So these two together are going to combine with NADP in this form, and we're going to add the electron. We're going to add the proton. And this is just going to form and then this, the important thing about this is that this is then going to go to the Calvin cycle. So when this takes it to another reaction, basically, this molecule is now going to transport our hydrogen to Calvin cycle, which is going to do the opposite reaction. This is going to take NADP, so the Calvin cycle of the light independent reaction is going to convert our NADPH. It's going to oxidize it back into NADP, and the NADP, it just doesn't disappear. The NADP comes back over here so that it can be reused again. It's not wasteful. It just, all the NADP is doing is basically moving these guys. In fact, if I want to do my chemistry properly, it would be then plus a hydrogen atom, which is the proton and the electron combined. So it gives this away. It can take it from here where it's not needed and give it somewhere else. So that's what all those coenzymes do. They transport the hydrogen. And it's a redox reaction, a reduction, oxidation. Whenever something is oxidized, the other thing is reduced, because oxidation is loss of electrons or hydrogen, and reduction is gain of electrons and hydrogen. Okay, so let's catch up on some of those comments. I did mention the monohybrid inheritance. I could go and do some of that. Let's get my board rubber here, which is a fancy scrubbing brush for the time being, because I haven't got a board rubber yet because my whiteboard is new. How does a neuromuscular junction compare to a normal synapse? That's a great question. Let me know if you want to do the monohybrid inheritance. I think, how does a neuromuscular junction compare to a regular synapse is a good question. Okay, so maybe I'll draw both of them. This is going to be a regular synapse. And this is going to be a neuromuscular junction. Okay, the distance is different. That's just because I drew it terribly. Okay, so a normal synapse, again, I'll run you through it in exam speak super quickly. Action potential arrives at the presynaptic knob. This opens calcium ion channels, which floods in. This causes vesicles of acetylcholine to fuse with the presynaptic membrane, releasing the acetylcholine into the synaptic cleft, which diffuses across, binds onto specific receptors on the postsynaptic membrane, which opens sodium ion channels. So the sodium ions then diffuse into this, which means it hits threshold, and we trigger an action potential in the postsynaptic knob. This is basically a regular synapse. A neuromuscular junction is a junction between a nerve and an effector. An effector is usually a muscle, could also be a gland, just as a little side point. Differences are, this neurotransmitter is usually acetylcholine, but can be dopamine, can be many other things. You only have to know acetylcholine as the example here. This is always acetylcholine. Capital A, capital C, lowercase h is shorthand for acetylcholine. So this is always acetylcholine. That's one difference. You also have more receptors on the postsynaptic membrane, because you always want a action potential to fire when you get some acetylcholine in here. This one, sometimes we do things called summation, spatial summation. There's also inhibitory synapses. You don't 100% of the time get an action potential here resulting in an action potential here. Not always. With this, 100% of the time, you get an action potential here, you get an action potential here, 100% of the time, and that's partly due to the fact there's more receptors. So if you release a little bit of acetylcholine in here, there's going to be more receptors saying, okay, I've got some acetylcholine, which will open up more sodium ion channels, which will facilitate the action potential in the postsynaptic knob. The other differences you'll see here is that you've got an enzyme in pits on the postsynaptic membrane. This is acetylcholine esterase. It's the enzyme that breaks down acetylcholine, because as soon as the action potential stops, you want the muscle to stop contracting. You don't want your muscles to be like, really slow to stop or really slow to start again. You want nice, snappy muscle reactions, basically. So in order to do that, you need to trigger an action potential super quickly because you've got more receptors, and you need to end an action potential really quickly, which is why you've got more enzyme. So you've got more enzyme in pits, they're in little recessed pits on the postsynaptic membrane. So the differences are, to answer your question, the neurotransmitter is always acetylcholine over here. Can be acetylcholine, can be other things in a regular synapse. You have more receptors on the postsynaptic membrane, which means that 100% of the time when you have an action potential here, have an action potential here, and you have pits on the postsynaptic membrane which contain the enzyme acetylcholine esterase, which break down the acetylcholine so it can diffuse back into the presynaptic knob and be reabsorbed and reassimilated. Okay, how far am I behind with the comments? Ronnie wants a Zoom link. I can do that, but I'm going to have to sit down. Oh, fine. Additional lighting. Okay, so let's monoclonal antibodies. Lots of year 2 topics coming up. I'd like to choose something that's a little bit more year 1. Let me copy my link and I don't even know how I do that. Oh. I've invited you, Ronnie. I think you'll have to check your inbox. Okay, what else? Monoclonal antibodies, I put them on the list. It's also, yeah, okay, so we could do that. Okay, before that we had Elevology, what are biotic and abiotic factors and examples of them? Please do answer this. Okay, young Ace, we can definitely do that. So biotic factors are all the living components of the ecosystem. All the bacteria, all the fungi, all the nematode worms, all the human beings, all of the things that are alive in the ecosystem. So the abiotic factors are all the things in the ecosystem that are not living. The wind, the rain, the altitude, the aspect, the inclination, the soil pH, the temperature, the yeah, all of the things which are not living, but obviously affect the way that things live there. So those factors which are alive are the biotic, bio meaning living, biology, the study of living things, and abiotic. A in biology just means, in science, it's Latin for not, atheist, asymmetrical, atypical. It means not typical, not symmetrical, or not believing in a supernatural God. So that is that enough, young Ace? And some examples, I gave you loads. That should be all good. Talk about tropism. I've done tropisms. Hey, Mitch. I often get called Mitch when I'm on the phone. I phone, what's your name? My name's Rich. Oh, hi, Mitch. It's like, there was definitely an R at the beginning of that. Okay, monoclonal antibodies. Aaron, you could give me a bit more of a question than just monoclonal antibodies, question mark, and bodies, just for reference, is IES, not YS on the end. Well, then why don't you jumped in and answered my abiotic questions before that? Can I explain what happens during hybridization, especially the part where chromosomes double? Thank you very much. Okay, so chromosomes, I'd say first word of warning, I didn't get my head around meiosis and genes and what was actually happening there when I did my A-levels, which caused me major upheavals when I went to university. But even if you're not, I didn't do very well on those parts of the exams because I never really understood what's happening. What is a homologous pair? Where are the genes? Why do we have 2 copies of the genes? What's the difference? Why do we have 46 chromosomes and 23 pairs? I didn't understand those things. And if you're going into the exam and you don't have a solid understanding of like those nuts and bolts basics, it's going to be really tricky to try and answer the questions on the new spec papers. So during cell cycle, classic combination, so a cell is going to basically multiply its organelles. It's going to add some mitochondria. It's going to add some ribosomes. It's going to increase its organelles. It's also going to duplicate its DNA. It's about to split in half. So it needs to double everything before it splits itself, so that the 2 copies are going to have the DNA that they need. Now, a regular cell, what we call a somatic cell, let's say a skin cell, a hair cell, an eye cell, all the cells of your body that are not your gametes, i.e. your eggs or your sperm, are somatic cells. They have 46 chromosomes, 23 of them came from your mum, 23 of them came from your dad. Each of those 23 code for the same thing. So chromosome 1 codes for whatever chromosome 1 codes for, and there's loads of genes on that. And your dad has a copy of this, and your mum has a copy of this. So you've actually got 2 copies of chromosome 1. And so that's a normal, regular, healthy somatic cell has 2 copies of each chromosome. Now you're going to double this. So now we're going to have 4 copies of every chromosome. You're going to have twice the amount of DNA before replication. And then after the replication, you're going to half that again. You're going to go back to 2 and 2. So that's where a lot of the confusion comes in, because there's the 2 chromosomes that you have naturally, and then the doubling effectively to make 4 chromosomes. During wheat hybridization, wheat's funny because it's a plant. You don't. What spec are you studying for wheat hybridization? I'd be kind of unusual, because plants have weird endosperms, and you can have triploid cells and all sorts of things in plant fertilization. It could be a WJC question. Just let me know, and I can finish answering that question so that you don't get too confused. How often are the live streams are throughout September: Monday, Tuesday, Wednesday, Thursday, 8 p.m. till around about 9 p.m. whenever we basically get bored of you. No, not really, but when we think we've run to a bit of an end. Do I have any abbreviations for the steps in the nitrogen cycle? No, my nitrogen cycle video has been watched nearly 100,000 times on YouTube, and I don't know how many times within my course itself. In terms of the abbreviations, make sure you are just, you know what you need to know. AQA students do not need to know the names of the bacteria. OCR students do need to know the names of the bacteria. AQA students don't need to know about Azotobacter and free soil living bacteria doing nitrogen fixation. OCR students do, so make sure you know what your spec tells you need to know. What is a niche from Hannah? A niche is, textbook definition for the exam, the role an organism plays in the environment. That doesn't really tell us what a niche is, but that's the answer they want on the exam. No 2 species occupy identical niches. Classic example of this would be like wading birds. If you take a sample of wading birds, they're going to be feeding for insects below the mud, typically, and they're going to have different length bills. Some of them have got tiny little bills and will be just scraping below the surface. Some of them are going to have super long bills that are going to be going down quite deep and getting the stuff that's deeper down. Some of them have a whole range of intermediate sized bills. If they all had a bill that was exactly the same length and they were all trying to eat exactly the same food, they'd be in direct competition for everything, all their food. And this is obviously not a great idea, because why fight for a resource that you don't need to fight for? So the niche is the exact set, everything it does in the ecosystem. So that's the places that it sleeps, the things that it eats, the things that eat it, exactly the role of a species in an ecosystem, like everything that it does and that is unique to it. You might have like great tits and blue tits, very similar niche, similar woodland, but they all eat slightly different things. If no 2 species can perfectly overlap, otherwise one of them will outcompete the other one, and one of them will go to extinction pretty quickly. Evaluate the importance of inorganic ions in biology for 25 marks. Okay, this is an essay question. I'm not going to go give you an hour-long answer to this, but think about what do. They are central molecules in things like enzymes, or the iron in hemoglobin, variable oxidation states. What other things do inorganic ions do? You've got magnesium, a central molecule in chlorophyll. So you'd be looking to try and pull in a wide range of the inorganic ions from a whole load of things. Phosphorus, what does phosphorus do? Well, you make ATP. Nitrates and nitrite ions, what do they do? So that ties back to the nitrogen cycle to make proteins, to make DNA. So you've got all these different examples of inorganic ions on where they fit into the biotic world, basically. So they're going from inorganic, from soluble molecules that basically get sucked up by plants, converted into molecules, and then eaten by animals, and then decomposed by bacteria or saprobionts. Interesting. Yeah, I can. Ronnie, I don't know without faffing around. All I can do is I can go to invite, and it says invite Ronnie, and I say yes. Oh, actually, okay, I can. Let me try this. I sent you a Slack. Okay, next question. I've now lost where I am, so people asking about what our ultimate guide use. So ultimate guide use, we have two basically. Normally throughout the year, we're going to be doing live teaching to our subscribers throughout September. We're just doing it for free. And like, these are super casual, kind of like not very well planned, just dropping in homework help questions. So there, yeah, our regular web classes are going to have like handouts and be super duper organized. And basically you're going to be able to be much smaller groups and all the rest of it. Do you have to pay for this month? You can join it absolutely free using the coupon Try It Free you can choose any of the courses from our Tailored Tutors website and just use the coupon Try It Free and it will make the first month for free. Yes, you do have to enter your bank details. Absolutely you do. But it just makes the first payment 0 and you can unsubscribe at any time. You can choose, yeah, Starter Guide or Ultimate Guide. Starter Guide is videos only, basically, and Ultimate Guide is all the videos, the resources, the questions that you can ask us, the web classes that we're going to be doing in October. They're the main differences. More questions for Ronnie. Okay. I'm just having math chat, just being hijacked by math chat. Okay, so what other, we can do monohybrid, dihybrid inheritance potentially. I'm sure I missed a couple of questions though, because I got, what's going on? Do we have a Ronnie over here? Oh, okay, I have to admit you. Allow to talk. And I can unmute you as well. Oh, you just muted yourself, I think. I think we're getting there. Ronnie is finally in the room. Hey, Ronnie.
Yay!
Yeah, that was a bit of a palaver.
Yeah, I didn't get any of the things you sent. But anyway, yes, I'm here now. Cool. So basically, yeah, carry on. I'm going to just shout the main chat things that I can't answer the questions of, okay?
Okay, yeah, no problem at all.
Cool. I'll catch up with where we got to. What were you last talking about?
I just finished a little while. I just re-explained what Starter and Ultimate were for a few people who were asking. I think more or less I'm up to speed. Did I miss any biology questions? If I've missed any biology questions, I had monoclonal antibodies as a question, but I'm going to make you give me a more detailed question than that. Just monoclonal antibodies question mark doesn't tell me what you need to know. It's quite a big topic.
There was a good comment. Sarah said, there was a comment from Caitlin, I think, yeah, about like, how do you remember things? There was a specific thing. How do you remember all the cell organelles? And Sarah had a really good idea, drawing cells off by heart and then rechecking and adding what you missed.
Yeah, definitely. So there's quite a few different examples. We'll need to know different ones, but all the core ones that you need to know are going to be things like mitochondria, chloroplasts. Obviously, they're quite similar in some ways. They are both the product of what's called endosymbiosis, which is they used to be free-living bacteria, and now they were basically enveloped into a bigger cell. And instead of just munching it up and eating that bacteria, it realized that it could come to a bit of an exchange. So the plants will give chloroplasts, they'll supply them with all the things that they need, and chloroplasts will take the light energy and convert it into sugars and then give the sugars back to the plant. So everything's kind of happy. It's kind of a bit of a win-win. As a result, they've got their own DNA and some funny things like that. What do you need to know? You don't need to know that much about them. You need to know 2 bullet points. Maybe mitochondria and chloroplasts are slight exceptions, but endoplasmic reticulum, Golgi apparatus, nucleus, ribosome. You need to know 2 bullet points about each of them, and that's it. All the textbooks will give you a paragraph on each of them. You need 2 bullet points. So check out the course videos to see exactly what those what the organelles you need to know for your specification are, and for what the 2 bullet points. Exactly the same way as mitosis, stages of mitosis. You need to know 2 defining features or 2 bullet points about prophase, 2 about metaphase, 2 about anaphase, and 2 about telophase. Often it's way overcomplicated in the books, but the exams will ask you for up to 2 bullet points per, or 2 factual points per stage, and to be able to recognize them. But that basically ties into those bullet points. That's that.
AJ's got a maths question, but I'm just saying we'll do it at the end, yeah, not now.
Yeah. You can keep your dirty maths out of my nice clean biology session for the time being. Yeah,
except for it's not my fault if the students bring maths in, Rich. They want to do the maths, they want to do.
That's their problem, isn't it? Although I am going to be, Ronnie's persuading me to do some maths at some point, so she's going to be teaching me some maths throughout the year.
Have I actually persuaded you now?
I don't think I'm going to do the full A level this year. I've got a little bit.
No, not that. No.
A little bit on my
just
plate.
Maybe a little bit, yeah.
I'll open the door to a bit of it, yeah, for sure.
Yeah, Caitlin's asking about how much it costs and what you kind of get for the ultimate guide, but Ryan's doing a brilliant job of fielding loads of questions because he knows all the answers.
I'm pretty on the payroll, mate.
Yeah, exactly.
So yeah, essentially it depends if you're year 12 or year 13. If you're year 12, it's £19 a month for the starter guide, or £39 a month for the ultimate guide. If you're a year 13 student, it's £29 for the starter guide and £49 a month for the ultimate guide. And
but that's for like basically a ridiculous amount of tuition.
You get a huge amount. Everyone, the common conception that people have is like, oh wow, that's really expensive. Well, I can tell you right now, the amount of time you get from us is, it's going to totally replace the need to have any kind of live tutor, which is going to cost you that per hour rather than per month. And you can use it as much as you possibly want. So yeah, you get, per month, you get 2 biology web classes per week, year 12 and year 13. And so there's, what's that, 8 per month. 8 classes with me in a small group per month, asking me all the questions that you need, plus all you can ask me questions on any of the videos 24/7, basically.
Biology question.
Yep.
I think it's biology. What is the Pacinian corpuscle?
Pacinian corpuscle.
Okay, that's what I said, yeah.
One of them. Okay, so there was also a question about past papers, which Sabrina asked. I'll just go, yeah, we can answer that after. What is it? I don't know exactly what the question is. You're just stuffing your face there. What's going on?
Yeah, no, it's pizza day. I've got pizza.
Okay, so the Pacinian corpuscle is a touch receptor. Every time you feel something touching you, it is because the Pacinian corpuscle is basically detecting it. You have, if you think of, it's quite hard to do that drawing. If basically the Pacinian corpuscle is a bit like concentric rings. You imagine a whole load of concentric rings, and it can detect, if you push on the outside of those concentric rings at some point, it's going to stretch them. And they have what's called stretch-mediated sodium ion channels. So when you stretch, because it's being bent, these membranes, these lamellae are being bent, it opens up the sodium ion channel. And if then you studied a bit about nerves, you know that if you release enough sodium into the axon, eventually you'll reach threshold, and then you'll trigger an action potential. So the Pacinian corpuscle basically has these special sodium ion channels that will open to allow sodium in, and that's proportional to how much they're stretched. So the stronger the touch, if a tiny, I'm sure lots of you, I use this example in my lesson on this, I'm sure you will have seen an insect, a tiny fly or mosquito land on yourself somewhere, and you can see it, but you can't feel it. You've got I literally can't feel it. And that's because your Pacinian corpuscles will be stretching a little bit to allow some sodium in, but they won't stretch enough to reach threshold. So you won't trigger any action potential. So literally your brain is not feeling it. Literally there's no signal going to your brain. You can see that it's on you, but you can't feel it. So the Pacinian corpuscle is a touch receptor, and it works by opening or closing a sodium ion channel depending on how much it's stretched. And obviously the harder the touch, the more it's stretched, the more likely it is to reach an action potential. It's a pressure receptor. Very well said, Sarah.
There's two really, well, one of them is a biology question about myosin-actin interaction and how it helps expand and contract muscles. But before you do that one, there's a really good question. There are so many definitions in biology. Do we need to know them all? Lord knows I haven't got time to learn everyone. And I know that is a big deal in biology, but you are going to use the weekly webinars to help people to keep remembering everything. You've got the side of the template note.
Accurate use of terminology in the exams, 100% essential. You cannot get good marks without accurately using the vocabulary. So in each of my lessons, I'm sure you'll just Google one, just YouTube it, just tailored tutors, whatever. And most of them will have, if they involve a key term, they will all have red key terms in there. They are the ones that you will need to know. It's not often you get asked, define what is meant by the term the. Not often you have to just regurgitate, very, very occasionally. And to be honest, if you do, it'll be 1 mark. And so probably not worth losing too much sleep over. But being able to use those words accurately is essential. So getting in the habit of just, instead of saying breaking down, you need to say hydrolysis reaction. Instead of saying joining together, you should be saying a condensation reaction. So getting in the habit of using them all the time means that they just become part of second nature. And yes, there's billions, but you only really need to know the ones that I've put in red in my videos. So you can make a little glossary of those key terms. Also, the new template notes we've done has a section for those key terms, so you can fold it over and test yourself on it, just like a flashcard, basically.
So those key terms in biology, it's not just sort of technical language, it's also that you can't use colloquial language like joining up and whatever you just said.
You can't, no.
Yeah, and that's part of what's on the left-hand side, is it? All of those, like what we would normally use as colloquial, but not.
Yeah. So
the fact that you don't get marks for it,
that you think you know, like population and community. Population has a very specific term in ecology. It's all the organisms of a single species in one place at one time. And a community is all the organisms of all of the species in one place at one time. So yes, you have to know, and you can't understand the question if you don't know what the words mean. So it is super duper essential. Lots of practice. And I mean, there are millions and millions and millions of key terms in biology, but you only have to know the ones that I've told you by putting them in red. Sabrina asked about downloading the past papers. In the past paper library, there's a link to a folder. You can download all the past papers in one go, which is super duper handy.
Rich, I'm wondering if it's worth like going on and looking at some of the comments on the biology course.
Yeah, I've got a couple. I can definitely...
Sabrina said she'd asked a couple and she's not sure because it says they're waiting to be reviewed.
What's your... Sabrina, you'd need to tell me your login name. I have Saba. The outstanding comments that I haven't replied to, there's maybe 7. There's Saba, Ibrahim, Saba again, George, Yassine, and Danielle, and Zakaria. They're the ones that I haven't answered. So I don't see any with the name. Could be Saba. Could be Sab-R, maybe.
Yeah.
So,
what are those questions?
The monohybrid and is monohybrid inheritance always 3-to-1 and dihybrid inheritance 3-to-1? I'm going to need to draw that. So if there's anything there that you can, let me just wipe my board and hand over to Ronnie for just a second.
Oh yeah, am I allowed to do some maths? Woo!
Quickly, yes.
Quickly, yes.
I'll set it up.
Okay.
I'll set up the full table so that I can actually do it.
Yeah, while you're setting up, I can't remember, whoever it was, type the question in again about the modulus. Basically, the modulus does not mean plus minus. You can't just, this is what students do. They remove the modulus sign and then they think that there's 2 versions of it. One with the modulus sign just kind of removed as if it was invisible, and the other one with the modulus sign replaced with a minus. That is not what the modulus does. You need to think of it in terms of graphs. So you've got a graph that is this shape because you're only going to have the modulus of a linear function. So it's going to be a V shape. And one of the branches of that is the positive part of the function. And the other branch is the negative part of the function. But algebraically, that negative part just continues all the way down to infinity. And the positive part continues all the way down to infinity. So if you say to the algebra, where does the positive part cross some other curve, and then you say to the algebra, where does the negative part cross some other curve, then it will tell you, but it might be chucking up some extra solutions down here that you don't want, because actually your modulus only exists down to my wrists. So you cannot treat modulus questions purely algebraically. You have to sketch the transformation of the modulus graph, have a look at it, and decide whether or not you want the positive or the negative part of the linear function, the plus or the minus. And if so, you solve that equation and you understand the solutions in the context of the graph. Because modulus graphs are really weird. They're basically two different functions, but only half of them, the domain restricted, so that you only get this bit of it and then this bit of it. So you just can't do it purely algebraically. I can't remember, whoever it was will have to type it back in because I can't remember the exact equation, but it was to do with it was the answer. Basically, you can't just whack plus minus on. Oh, Rich, you've showed me this. This is the thing with the probability or something.
There is some probability involved in this. I can't see the comments, so just shout if.
Yeah, I will. Yeah.
And am I light enough? Is it visible?
Yeah, that's good.
I'm lighting out in here, but I wonder if this is going to help actually. Is it going to be any more light, any less light?
No, it's fine. We can see it. You've got a shadow, but it's fine. We can see the board really clearly.
So the thing is with this that students always get confused on is like, how do I know? Surely there's more combinations, which is basically the question from Sabrina. I assume it's Sabrina. This is mono hybrid, which means you're looking at one gene. So the two alleles, we've got the tall gene, it's dominant, big T, and the little t is the dwarf gene. And so we're going to start with one parent is going to be big T, big T. They're always going to set you off with big T versus little t. So what possible choice does this organism have to make gametes from? It's only got big T's as its choice. So each of its gamete is going to be haploid. So these are going to be separate, but one of them, the only choices that they can be are big T. So the blue parent produces the gametes, big T, big T. The little t is going to produce these gametes. And then we're going to do a cross. So whenever you don't really have to do this once you've done a few of them. Whenever you see parents which are homozygous with homozygous, you're going to get a 100% heterozygous in the next generation. So we can do this: big T, little t, little t. So all the offspring are going to have this genotype. So that's the first step. We haven't started with the cross yet. So now we're going to cross these with each other. So they're going to inbreed this is called the F1 generation. So what are we going to do? We're going to have one parent is going to be this, and the other parent is going to be this. What gametes can they make? Well, this one is going to make one gamete like this, one gamete like this. Same for the other one, because they're both identical. So these are our possible gametes. So we're going to cross a big T, little t with a big T, little t. Okay, so what are we going to get? We're going to get big T, and we're going to get a big T and a little t. Going to get a big T and a little t, and a little t and a little t. So now they can ask you two things here. They can ask you for the genotypes of the offspring, which was not the question. And then the phenotypes. Okay, the genotypes. Well, this is what's going on with the genes. So I'll do them in fact, now I'm going to highlight that color in there. So let's just we're going to have one of these. We're going to have 2 times big T, little t, and we're going to have one of these. The question from Sabrina, though, was, is the phenotypic ratio always 3:1? Okay, so what's the phenotype of this going to be? Well, it's going to be big T. If it's big T, little t, the dominant allele will come out in the phenotype. So these two are also going to be tall. So I'll put the genotypes here. Tall, tall. I'm going to write tall again just because we've got two of these. And then we've got dwarf. So the phenotype ratio is 1 to 3 to 1. 3 tall to 1 dwarf. So I started over here because students always get confused about how do I know I start with this, because this is the starting point always. And this is the starting point always because these genetic experiments always start with a homozygous dominant crossed with a homozygous recessive to produce 100% heterozygous offspring in the F1 generation. And this would then be called the F2 generation. And the genotypes are going to be 1 to 2 to 1, and the phenotypes are going to be 3 to 1 always. So that is a monohybrid cross, and that's why you get in normal circumstances a 3 to 1 phenotypic ratio. Now the question was, do they always result in a phenotypic ratio of 3 to 1? Unless something very unusual happens, and in monohybrid, basically, it's always going to be 3 to 1. I've never seen questions that ask you unusual monohybrid. These are going to be 1 or 2 mark questions because they're very straightforward to go through. You don't normally have to do this bit. I'm just showing you this bit because otherwise you get confused as to why do I start here? Because this isn't really because you could start with any combination. Okay, so that is that. I do dihybrid as well. Dihybrid is a bit more complicated.
And then can you explain the difference between genotypes and phenotypes?
I will probably do that first because that's a little bit essential. So, whilst I've got a nice whiteboard, we've got the tall allele is T. So the genotype, this is what's called, there's lots of terminology here. This ties in nicely to our key terms. And yes, you do need to know how to use all of these, not necessarily how to define them, but you have to be clear in your understanding. This we would call heterozygous. Heterozygous means it has 2 alleles which are not the same as each other. If it's this, or this big T, or little t, this is homozygous. It has 1 allele only. Homo the same, zygous, and this is hetero. Okay, this is the genotype tells us exactly which alleles it has. But the phenotype, what does this look like? If we put this into a plant and grow it, it's going to have the phenotype of the dominant allele. So the phenotype would just be tall. I'll do these separately. It's going to get in the way, so let's just replace them. So this guy's going to be tall. This guy is going to be tall. And this guy's going to be a dwarf. So the phenotype is, what does it actually look like? What does it actually grow up to be like? And that's going to be, if it's heterozygous, it's going to be the same as the dominant allele. The only way it can grow up to be a dwarf is to be homozygous recessive. It needs to have both recessive alleles. Okay, that's a 101 on what those words mean. Let's see how quickly I can do this without making mistakes. Okay, so we're going to start again. Oh, we're going to need 2 genes. We're going to need T. What's a good one? TTEG. So here I've got, this is a dihybrid cross. So I'm using 2 letters because we're looking at 2 different genes. We've got T and G. And again, you're always going to start with homozygous dominant crossed with homozygous recessive. and that's going to produce 100% heterozygotes in the F1 generation. That's just, if you want, you can draw it all out for yourself. I'm not going to go through the whole entire thing, but I'm going to end up with heterozygous for T, heterozygous for G, and we'll have basically all of them will be big T, big G, little g. That's how the F1 generation is going to be. Have I lost anyone at that point, Ronnie? Is there like...
Yeah, no, everyone. In fact, the opposite. Caitlin is saying it finally makes sense.
Okay, great. So this is not a very well drawn T.
And Seb says it's his favourite topic apart from domains and kingdoms. But Hussein says it's his least favourite topic.
Yeah, it's a bit divisive, this one. Once you get your head around it, it's not too bad. Okay, so we're going to cross this with itself. And this is the cross that's going to result in a 93 to 1. Okay, so we're going to have, what gametes will this make? This is going to make, we're going to now have the gametes. What color do I want to do this in? Oh, I'm going to stick with black for the time being. So do I want to separate them? I'm going to draw my table. How low can I go? Yeah, I can fit my writing in underneath. Okay, maybe I can draw this. This is where a lot of students go wrong. How can I draw this?
Oh, you can go further down, further down.
Yeah, no, I'm going to need to put stuff down here. So that's why I've moved that space. So I'm using my brain a bit here, Ronnie. Okay, so these are going to be on separate.
What are the lines?
These are chromosomes.
Oh.
And this is the position of the T allele. Maybe I'll do my G's in green. Maybe that's going to... Okay, so this, in fact, let's not do it in the same spot. If they're on the same chromosome, they behave differently. That is autosomal linkage, and that's a whole other topic right there. But the reason these are on separate chromosomes. So what's the possible different ways these can be inherited when you do meiosis, when you make your gametes? Yeah. So when we make our gametes, they're going to be haploid. They're only going to have one T, and they're only going to have one G. So we could have big T, big G. Well, is a possible combination, right? Another possible combination would be the big T. Ronnie, can you help me?
I was just talking to AJ. Big T, what? Big T, little T?
So we're gonna the gametes that these guys are gonna produce are gonna have one T and one G, and they can have any combination of those big. So they've got one big T, yeah? And now here I've done the big T with the big G. That's this one here, this one's done. So we've got this big T can also combine with the little g, and this is
the little T with the big G or the little T with the little g.
You got it. So these are the only possible combinations of what the gametes are going to have, right? Are you with me on that, Bailey?
Yeah.
Okay, and again, we're going to cross this with ourselves. So let's do.
Yeah.
Sorry. I think I'll move these. I don't think it's clever to get rid of them, but I'm just going to move them over here. Okay, and then I've got G, G, G. And exactly the same, you're crossing it with itself. So whatever goes on the top here also goes down the side here. Give yourself some room when you're drawing these out because it can get a bit clustered. I like to draw my gametes in circles because it just differentiates them with what's going on elsewhere. Okay. Now it's just filling the gaps, right? So we're going to have Big T, big T.
Wait, is this like mixing a T, capital G with a capital T, capital G, and then mixing
Yeah, so
it
this is going to be if this gamete mixes with this gamete, what's the offspring going to have? Because remember the parents, everyone's a diploid. All the normal human beings have 2 copies of the T and 2 copies of the G.
Okay.
And when we do meiosis to make gametes, you need to half that. So you basically will just get rid of these. So you could have this G and this G, you could have this one, and you could have this one and this one. So the combinations that you make in your gametes, that's meiosis, that basically means you have any one of these possible combinations is possible.
Yeah.
But some of the combinations are more likely to happen than others, as we're about to find out. So I'm taking the big T. In fact, let me just put the big T in, yeah, for this guy down here. And I'm going to do the same big T for this guy down here. And then I'll do all the little T's. Oh, actually, I'll do them in a slightly different order because it will be confusing otherwise. Okay, now I'll just do this big T with this big T, this big T, this big T, this little T here, this little T here, this little T here, this little T here. Same again. And now I'll do my big T. It's always best to write the dominant allele first. Ronnie, can you tell me what T's going here? I know you're doing other things at the same time. So I'm taking my T from here and my T from here. What's my 2 T's, big or little?
What, down there?
So what's this T?
Yeah, little. Small.
Little. Okay, and then this one would be, what's the T's?
Small.
Top row.
Top row would be big, small.
Big, small.
Yeah, big, small there, and then big, small again, and then small, and then small, going down.
Okay, now I'm going to do my G's in much the same way. So I've got big G. I've got big G, little g. I've got big G. I've got big G,
big,
G.
little.
And then little.
Does the order matter?
You normally put the dominant allele first. It doesn't really matter, no, but it's just good practice.
Okay, so the next one's little big, and then little. And then big, and then big, small, and then big, and then small. And then big, small, big. I can't see your head's in the way.
Yeah, big, small, and
small. Yeah.
Okay, so whenever you've got a dominant allele, it will be expressed in the phenotype. So let's look at...
But most of those are the same.
A lot of them are the same. Yeah, and this is where we're going to come with our 9 to 3 to 3 to 1. So let's do the genotypes first. I'm going to need some room for this. Okay, so genotypes. We're going to have, I'm going to just swap. I'm just going to do
is Osman saying, is it the same vertically and horizontally? I didn't even notice that.
Yeah,
yeah.
Itself. So this is this, you're crossing it to self-cross. So yes, what goes here also goes here. Otherwise, it doesn't work. Okay, so we've got big T, big G, big G. So we've got one of them. Do we have any more of them? I don't think we do we? We're gonna have one of these. Yeah.
Caitlin's saying, what do all these combinations mean? Are they the potential gamete offspring produced?
These are gametes. These are the potential offspring, possible combinations of genes in the offspring. Yeah, which hopefully will become clear in just a moment. So we're now looking for big T. I've done big T, big G's. So I'm going to do big T, big T, little g. Okay, so I've got one here, 2. I think that's all the ones I've got, yeah? So these are done. And then I'm going to do, what other big T's can I do? I can do big T, little g. Okay, so how many of them have I got? I've got one here. Big T's and little g's. I think that's going to be the only place. So one of them.
Yeah, because there's only one way to choose that.
Yeah, this one and this one.
Yeah, there's only one way.
So now I've done all my big T possible combinations, yeah? So now I'm going to do my little t combinations. sorry, my heterozygous combinations. So I could have this, big T, little t, big G, yeah? So that's gonna be, I'm gonna need little t's, and there's gonna be one of them, yeah? With me?
Yeah, so the second position is gonna be a small t regardless of what, yeah.
Oh, in fact, no, that's totally wrong. Totally not what I've written down. I've written down big T, little d. So I'm now looking for big T, little t.
Or little t, little t.
Yeah, that one was wrong. So we've got this one here. You basically don't ever have to do this in the exam. I'm just trying to show it all completely laid out.
And then you could have big T, little t, big G, small g.
Yeah, exactly.
And then you could have, little t, big G, small g, little t.
We also haven't done this one. Big T, little g, little g. And then we can have the little t's only, so little t's with big G's, yeah?
Yeah. But what are you actually doing?
These are all the different possible combinations of genotypes. So they don't ask this in the exam. This is like beyond, because this takes too long. You can see by me doing it, it takes too long. These are the possible genotypes. But what are the phenotypes? This is the question, is what are the phenotypes? Okay, so the possible phenotypes you're looking for, if it's got a single big T, one, doesn't matter if it's got a big and big or a big and a small. If it's got a single big T, it will be tall. Its phenotype will be tall because it's a dominant allele, right? So so anything with a big T is gonna be tall. So all of these guys are tall, yeah? But there's two qualities here we're looking for. Let's say G stands for green, yeah? And big G stands for green, and little g stands for red. Happy with that? Big G's are green. You're not talking to me, Ronnie.
Sorry, I was miles away. I was reading. Fatima is doing a really good explanation, which I was reading and trying to understand, but I think this is beyond me. But yes, big G is green, small g is red. Like hair, yeah? Red, green hair, red hair.
Okay, so now let's find, it's easiest, a little tip here, always start with the recessive ones because they're going to be the easiest ones to find. So what would be the phenotype, Ronnie, of this guy here? This is the weird one. This is the odd one out. It's little t, little t. Is it tall or is it dwarf? You can ask me questions if you don't know.
It's got red hair.
It's got red hair, and the T?
It's small.
It's small, yeah? So both small ones and both small ones, yeah? So this one.
Small, red.
Exactly. So the phenotype is only, that's the genotype. The phenotype is small and red, dwarf and red. Yeah? And that's the only one in this whole grid of 16 that will be that will look that way.
Yeah. Oh, so yeah, because it's recessive.
Now I'm looking for other double G's. Where's my little g?
Along the bottom row.
Yeah, exactly.
No, not along the bottom row. There's one there, and then there's one to the left, and there's one two up. Yeah. Yeah. And left two from there.
Yeah, okay. Yeah, let's actually take this guy out. Because now I've got big T.
Wait, that one's wrong, Rich.
No, don't worry, we're good.
No, the one that is the, put your finger on between the red and the green at the bottom. Yeah, shouldn't that be a capital G?
Should be a capital G.
Yeah.
Either way, it's wrong. So now I've got a big T, one big T, and two little G's. This means he's going to be tall. And what color?
Red.
Yeah. Okay. Now I'm looking for, can you find me any that are going to be dwarf and green? So what am I looking for to see a dwarf? What do they have to be a dwarf?
Small t, small t.
Small t, small t. And what do they need to be green?
Big G, small g, or big G, big G.
Yeah, any big G, one big G at least. So we've got 1, 2, 3. Very confusingly, I labeled my dwarf in red and green pen, but there we go. And then all of the others have got at least one big T and at least one big G. So what are they going to look like? If they've got a big T, they're going to be
tall and green.
Tall and green. So we can count them if we want to. 1, 2, 3, 4, 5, 6, 7, 8, 9. So if we have tall and green.
Rich, you going to come on Thursday and answer my maths questions?
I can try. Yeah, for sure. So the ratio, we have 9 tall and green. We have 3 dwarf and green. We have 3 tall and red, and we have 1 dwarf and red. So the phenotypic ratio is 9 to 3 to 3 to 1, because you are 9 times more likely to get this combination, tall and green, than you are to get, there's only one possible combination of fusing of gametes to produce little t, little t, little g, little g. And therefore you're much less likely to get this. You're 9 times, in fact, 9 times less likely to get this combination than the combinations that make up tall and green, which is any single big T with any single big G. And that
does this carry on and carry on? So like, for example, say, you know, in the bottom right, you said they are dwarf and red. Yeah. Does that mean that they are essentially small t, small g? So like say a TTGG then has a child with another one, like can you go back to it being 2 and 2, or is it now 4 and 4? And you've got to like consider.
No, okay, so like this is the parent. Yeah, this is the new organism, which is diploid. It has 2 of each. When it wants to make its own gametes, when it's going to go through meiosis, it's going to have 1 T. But what it's only got a choice of little t and little t. So its gamete, it can only make 1 gamete. This one.
Okay. Yeah. So you don't need to like keep getting like 2 to the power of combinations. No. It literally is just
with. If this breeds with itself, every single one of them
will be dwarf red,
because you've got no other choice. There's no other diversity. If you cross this with this, you'll get something else. This will not, the 9 to 3 to 3 to 1 will only apply in this circumstance when you've got this is the parent and these are crossed with themselves. They will only ask you the question in respect to that. The problems and the complications come to this were like if the G and the T are on the same chromosome, suddenly you fuck up these. These will no longer be the same, and that's when you would get unusual combinations. Basically, you'll get the same combinations what the parents are, unless some crossing over has taken place, and there's more complications to come to that. But understanding and just being able to draw this out for yourself and being really careful. Like here, I put the G in wrong, didn't I? I was just doing it quickly and I just put the wrong G in this column. Just a simple reading error. Like in maths, like you say, you're finding those errors.
All the time, all the time.
That would cost me the mark, or I wouldn't have been able to prove that I get my 9 to 3 to 3 to 1 ratio using that method. Because
so many like students always kind of think that in order to go up a grade, they need to learn more or understand more. Actually, they don't. What they need to do is just not make simple mistakes or be aware of
wording, usually.
And just be aware of how easy it is to make these little mistakes, and that then you know, you're going to lose grades, not because you don't know it, not because you don't understand it, but just because we all do silly little, we think capital G, we write small g.
Yeah, I was doing, I was probably talking to the camera and just like wrote it down.
Exactly.
So that, and if you're having trouble with this, draw it out from as I've drawn it, and then just fill it in and then highlight. Basically, it's easiest to start with the recessives, and you're looking for any of the times when you get little g's together. These are the easy things. And actually the pattern, if you lay it out the same way, is always the same. It's always this box, and this box, which have the same phenotype, and then these guys which have the same phenotype. So it's always basically, and then this guy in the corner is the totally weird one. But basically,
and the top left is weird as well.
Yes, but the phenotype of this.
Oh yeah, because that's I get you, because that's dominant. So the
to look at the plant, he would look exactly the same as this one, as this one, as this one. These all look identical. They have different genes, but the phenotype, what they actually look like, is the same. So yes, genetically speaking, it's unique, but in terms of the physical appearance, it's exactly the same as the other 8 to make the total of 9.
I think we should wrap up soon. Farhan is saying, can you cancel a subscription or is it fixed? Yeah, of course, you can cancel whenever you want.
Just like Spotify. Spotify. Start it when you want, stop it when you want. If you want the money guarantee to apply, you will need to be enrolled in the course for for the money back guarantee to apply, you need to have completed 90%.
Oh, but that's like right at the end, yeah. That's going up a grade. I think we're talking about like literally just now. If you sign up, can you? Yeah. And you don't like it, can you cancel it? Yes, of course you can.
Yeah.
Is there any more questions? Biology questions, TT, organisation questions, course questions? Anybody wants to ask before tomorrow? Tomorrow is Other Rich, as I call him, doing chemistry. And then Thursday is me doing some maths, and we're going to do some modular stuff because that is quite tricky. It's quite hard to get your head around. So definitely going to do some modular stuff and probably some quadratics, maybe a bit of discriminant stuff.
Okay, so Ronnie can teach me some stuff over Thursday.
Insulin production.
Insulin production. It's a hormone. It's made by. Is that just the question? Insulin production. I think we're here often enough. We'll save it up. If you have a question about insulin production, save it up for next Tuesday and write me a full question about insulin production, or
go to the website, use the try it free coupon.
Yeah, and there's
that means you get, and then you can just literally type your insulin production question into the insulin production video, and Rich will get back to you.
100% true.
Cool.
Okay, I think that's everything. Someone just said that the chemistry course isn't quite complete. No, it's absolutely not quite complete, but it is.
Yeah, it's more complete than my course, though.
Yeah, most of the Richard's videos are with the editor now, so they're going.
Yeah, and I'm getting there.
By the end of the month. I mean, you can try it out for free right now, so check it out, and it's for free. And by the time it's not free anymore, the course will be complete. It'll just be
Sugar's saying, what are our degrees in? Can you guess?
Yeah, biology and maths. Yeah, and although Richard actually, his degree, Other Rich is pharmacology, so he's a pharmacologist technically, but obviously it's so much organic chemistry and pharmacology. He's been teaching chemistry for years. Okay, wonderful.
Thank you, Caitlin.
Thanks for being my assistant today, Ronnie. And we'll come back next week. Will we be here tomorrow?
Yeah, and Caitlin, next Tuesday. But actually, that's the last one, isn't it? That's free.
No, we got next week.
Yeah, that's what I mean. But next week's the last one. Next Tuesday is the last.
Yeah, Tuesday's the last free drop-in
one, session. And then it will just be for the ultimate users. And Ryan saying, have fun marking. Thanks. I actually do like it. I quite like marking A-level papers. I'm marking remarks at the moment, so it's people who've put in for a remark. So yeah, that's what I'm going to be doing tonight.
Perfect. Okay. Thanks, Ronnie. I'll shut things off for the time being.
Yeah. See you later.
You tomorrow or later.
Yeah. Bye.